All right, my name is Savannah Collins-Key. I am a PhD student at the University of Tennessee in Knoxville. This research is based on the work that the Quantitative Wood Anatomy group performed last year at NADEF in Cody, Wyoming. Since last summer, two other members of the group, April Kaiser and Sarah Germain, and I have been working with our group leaders, Dr. Georg von Arx and Dr. Matt Becker, to continue this research and expand it. That includes continuing to process samples that Dr. von Arx has been working on over in Switzerland. So this is an expansion of a lot of things we wanted to do last summer, but couldn't quite get to yet. And it is split between the ideas. If you were at NADEF last year, we were hoping with this research to be able to look at several aspects of our data set. Sorry, I can't get it to go forward. Oh, there it goes, sorry. All right, sorry, first time on Zoom doing a presentation. So there are several aspects of this research that are unique that make us very excited about being able to learn with Dr. Becker and Dr. von Arx. Working with quantitative wood anatomy or dendro-anatomy allows us to look at the biological aspects of the tree and the tree's mechanisms for growing and survival, better than being able to look at it just visually eyeballing it or looking through a microscope or even scanning. Through the process of being able to take with a microtome slices and then being able to stain the wood samples, we can get this very fundamental picture of what the tree is doing and how it functions and the mechanisms that could be going into why certain trees are surviving and why certain trees may not be. So we asked a few questions last year, and we're still looking at now. Mostly we wanted to look at with our data set why certain trees survived and why others did not. I'll talk about our plot site in a moment, but these were all from the same plot. So we want to look at whether there were predispositions that certain trees might have had that would have enabled them to be able to survive or perhaps hindered their survival. And part of that is looking at how trees respond to extreme events. A lot of times the types of weather and climate events that would impact the trees at the elevation, the location that they are at, would be detectable within the tree ring. Sometimes not. You don't need to use a microtome and slide and be able to take the stain process route that we've been taking. But it makes it much easier to, sorry, I also have a dog that is making a lot of noise at this moment. But being able to take this route by staining and looking at the biological mechanisms of the tree ring and the anatomy allows us to look at these responses much more clearly and see aspects or features that we might not see if we were looking in the more traditional ways that we evaluate tree rings. There are several parameters we wanted to analyze having to do with tree growth and water relationships. This aspect of the research was mostly focused on vigor or damage. So a climate or weather event that could have occurred that would have caused mechanical visible damage within the tree ring, or perhaps a mechanical damage that occurred due to a geomorphological mass movement. The aspects of the tree that we'd be looking at to determine this type of damage would be resin ducts, frost rings and blue rings primarily, as well as several other aspects. But those are the ones that I'm focusing on today and that we're focused on the most. If you've never looked at these images before or seen what a frost ring or a traumatic resin duct looks like, these are basically what they would look like with the staining process that we use for wood anatomy. I don't know if you can see my cursor, I think you can. This image here actually shows all three of them in one. We have traumatic resin ducts down here, which would be a vigorous appearance or frequency of resin ducts. Blue rings, which just represent, they occur in the late wood, they represent an unlignified portion of the tree ring. So the lignification process never completed itself during the end of that annual rings formation. And then also frost rings, which usually occur along with blue rings and traumatic resin ducts. So we wanted to look at, because of the species we were looking at, which is whitebark pine, we wanted to be able to look at the resin ducts not just as resin ducts, but be able to quantify them at different levels. And we used DeRose and Becker and Long's paper as our basis for categorizing these resin ducts that we were observing in our samples. In their paper, they have actually level 0, which is technically just a constitutive resin duct, or the baseline appearance of resin duct in the tree ring. But we just assigned levels 1, 2, and 3 to resin ducts that we observed. Level 1 would look like this, and part of this categorization is that it can be subjective. So it depends on sometimes who's looking at it and what they believe they're seeing. Dr. Becker and I primarily were the ones who were classifying these resin ducts. I was trying to take Dr. Becker's lead on this. And we've been discussing in our group expanding that as well, so that we are following up on each other's determinations, but at the same time trying to make sure that there's consistency when we are judging these features. But traditionally, a level 1 traumatic resin duct will have a tangential alignment, some, but no grouping, no clear grouping. I tended to see if I saw at least like 3 or 4 resin ducts lining up tangentially, I would count that as a level 1 resin duct or traumatic resin duct, even though technically it's level 1 resin ducts aren't going to usually be considered traumatic. Level 2, you're going to have that same alignment, but much more grouping. And with level 3, the grouping, the alignment, and also a much more compact, higher frequency. And there are, at least in our data set with our species, there are very few examples of a level 3 traumatic resin duct occurrence. Other distinct features that we were looking for in this data set were what we believed were high wind events, which would occur often in the plot that we took our samples from. These could also be confused, again, subjectivity, with a frost ring. Technically, you don't usually see a frost ring in this part of the ring, which is why we thought that it might actually be something other than a frost ring that impacted the tree. Also scars, which actually there are very few of up there. I think we only had 2 rings with scars on them. And compression wood, which was usually more present in the earlier stages of the tree's lifetime. So this was where our plot was, outside of Cody, Wyoming, where where native has been held the last 3 years, I believe. Our sample site, all of our trees were found or were taken from the same site. So we don't have a variety of like geographical differences that could be influencing the tree. Total, and last summer we only had 10, but we've been able to process up to 15 trees now. 8 were alive when samples were taken, 7 were dead. Reaching back to 1868, we've counted over 10,000 resin ducts, 4.5, that's a guesstimate, million cells. All of these are processed using Roxas, which Dr. Von Arx developed. So just to wrap up, these are the initial indications we've gotten just from looking at the distinct features and what we hope are features that are caused by a disturbance. What we're hoping to do is be able to determine whether or not mortality or susceptibility can be determined from these features without being combined with climate, although we are also going to do that as well, but we thought we would separate them and see what we could get or divulge from the data set just by itself looking at these features. I looked at resin ducts, all levels, so level 0, which would be the CRDs, and then levels 1, 2, and 3 of the traumatic resin ducts, and compared all of the trees to the living trees and the dead trees. And we saw this initially, that there are quite a few more resin ducts in the trees that died before we sampled them than the trees that were still living, in all aspects, with all resin ducts, CRDs, and TRDs. And all of them showed mostly a downward trend, not much of an upward trend towards the end of the lifetime or in the later years or later rings. But again, when looking at traumatic resin ducts, the level 1 resin ducts aren't usually considered traumatic. So I looked again at just the resin ducts that were at level 2 and 3 only. And there was a little bit of a trend. Actually, everyone's faces is right here. I can't see. No. Yeah. There's a little bit of an upward tick with the dead trees when looking at the more traumatic resin ducts. So we ran a quick Mann-Kendall trend test just to look at p-values and see if there were trends. There were no trends with the living trees, but there was a significant trend with the dead trees. I split this up, which I left out of here to save time. But there was a significant, again, upward trend just in from the last 70 years, 60 to 70 years, depending on when the tree died. So I did look at other features just for this talk specifically, even though we're going to dive into that a little bit more thoroughly later on. There were not any trends that were discernible initially with frost rings, blue rings, or any other distinct features. A lot of this mostly just has to do with the fact that there just aren't there's not a lot of data. We're really proud of the data that we processed and the data set that we have now. It's a strong data set, but with just looking at the discrete features, I think it breaks down the data set a little bit too much to where we still don't have enough of those blue rings or frost rings. I think blue rings, we had about 50 or 60 blue rings that we saw, and frost rings about like 85. So still not too much, especially when you then start breaking that down into live and dead trees, enough to really be able to discern why a tree is surviving while another tree next to it isn't. But with the traumatic resin ducts, hopefully we'll have something, and definitely when we combine it with our climate data later, we'll be able to get a lot of answers we've been looking for since last summer. So just wanted to thank everyone in my group again, and Georg and Matt. If anyone has any questions.
Awesome. So there's questions coming in. We have time for maybe one question or so here. So the question is, do all of the chronologies go back to the pith? If not, when aligned by approximate age or maybe size, what does that pattern look like?
Ooh, that would be a fun question. I do think that the majority of them do go back to the pith, if I can remember correctly. If I'm wrong, I will find out in an email in the next few minutes. But I don't know if we have looked at it from that respect yet, but that's something that would be worth looking into.